This video is about the review of the lipid chemistry and classification of the lipids. Lipids are organic substances which are relatively insoluble in water but soluble in organic solvents like chloroform, ether, and benzene. Lipids are basically classified into three categories: simple lipids, complex lipids, and derived lipids. Simple lipids are basically esters of fatty acids with various alcohols. These are further classified into neutral fats or oils and waxes. In case of neutral fats, the alcohol is glycerol, while in case of waxes, the alcohol is other than glycerol, which is basically higher molecular weight alcohol. So the basic difference between the neutral fats and waxes is that in case of neutral fats, the alcohol is glycerol, and in case of waxes, the alcohol is other than glycerol, which is higher molecular weight alcohol. So the this is the structure of the glycerol. Here you can see that the glycerol, it is esterified with the three fatty acids. The fatty acids, they are represented as this R prime, our second fatty acids 2 prime, and third with 3 prime, and the alcohol in this case is the glycerol. These are basically uncharged molecules. This shows the esterification of different fatty acids with the glycerol. If one fatty acid is esterified, the glycerol is the monoacyl glycerol. If two fatty acids are esterified, the glycerol is diacyl glycerol. If three fatty acids are esterified, then glycerol is triacyl glycerol. So you can see here the backbone is of glycerol, to which three acyl groups are esterified. Waxes are basically esters of fatty acids with high molecular weight alcohol. Examples are lanolin, beeswax, and whale sperm oil. Complex lipids, these are esters of fatty acids with alcohol, but in addition to it, an other additional group is present, which is usually called as prosthetic group. So if the prosthetic group is phosphate, the complex lipid is called phospholipid. If it is carbohydrate, then it is called glycolipids. If it is protein, then it is called lipoprotein. So complex lipids are basically further classified on the basis of the prosthetic group. Phospholipids. We define phospholipids as that these are esters of fatty acids with alcohol with an additional prosthetic group in the form of phosphate or phosphoric acid is present on it. So here you can see that the backbone is of glycerol, to which two fatty acids are esterified and a phosphate group is present. Now to this phosphate group, an additional alcohol is present, on the basis of which we further classified the phospholipids. So they frequently have nitrogen containing bases. Now, what are phospholipids? How can we classify them? Already we have defined that phospholipids, these are esters of fatty acids with alcohol along with phosphate group present in it as a prosthetic group. So on the basis of the backbone alcohol, which is the main alcohol, on the basis of the backbone alcohol, they are further classified as glycerophospholipid if the backbone is of glycerol, or sphingophospholipid if the backbone is of sphingosine. Now here you can see that in case of glycerophospholipid, glycerol is further esterified to two fatty acids along with an additional phosphate group, to which alcohol is present. Now this alcohol may be choline, this alcohol may be inositol, maybe any alcohol is present. On the basis of that alcohol, we further classified that glycerophospholipid. But in case of sphingophospholipid, backbone is of sphingosine, to which fatty acid is esterified, phosphate group is present as an additional group. To that phosphate group, only the choline is present, not other type of alcohol. Only choline will be attached to the phosphate group. So this is the main difference between glycerophospholipids and sphingophospholipids. So phospholipids may be classified on the basis of type of alcohol present. So the types of glycerophospholipid, main backbone is of glycerol, and if alcohol present to the phosphate group is choline, that it is called phosphatidylcholine. You know that two phosphate groups are esterified to the glycerol, so it is phosphatidyl, and choline is present to the phosphate group, so it is phosphatidylcholine. If alcohol is ethanolamine, then it is phosphatidyl same phosphatidylserine, phosphatidylinositol, plasmalogens, and cardiolipins. In case of the sphingophospholipid, the main alcohol is of sphingosine, and the major type of the sphingophospholipid is sphingomyelins, which is abundantly present in the nervous tissue. So this is the biochemical structure of the phosphatidylcholine. You can see here that this is the glycerol, main glycerol, to which two fatty acids are esterified. To glycerol, phosphate is attached, and then choline is present. So this is phosphatidylcholine, and general biochemical name of it is lecithin, and it is present in the cell membrane. Similarly, you can see phosphatidylethanolamine, phosphatidylcholine, dependent that which type of alcohol is present in it. If it is ethanolamine, that it is phosphatidylethanolamine. If it is choline, then it is phosphatidylcholine. If no other alcohol is present to the phosphate group, then it is phosphatidic acid. And phosphatidic acid is the most simplest fatty acid. Similarly, if alcohol is serine attached to the phosphate group, then phosphatidylserine. If it is inositol, then it is phosphatidylinositol. This is the biochemical structure of sphingophospholipid. The backbone is of sphingosine, to which one fatty acid is esterified, phosphate is present as an additional group, and 2-phosphate choline is present. So this is the general structure of the sphingophospholipid. Glycolipids. The most important message in case of glycolipid is that they do not contain phosphate group. So if you have any MCQ like question which shows or which is written in it that phosphate group is present in glycolipid, then it is wrong. Phosphate group is not present in glycolipid. How can we define the glycolipids? These are esters of fatty acids with alcohol. Alcohol is sphingosine. A carbohydrate group is present as an additional group or prosthetic group with nitrogenous base. So examples are cerebrosides and gangliosides. So here you can see the structure of the different type of glycolipids. Ceramides, in which backbone is of sphingosine, to which acyl residue or fatty acyl group is esterified, and here this circle, which is slightly pink, it shows the attachment of the carbohydrate. Now in case of glycolipids, major carbohydrate or usually present carbohydrate is galactose, but glucose may also be present, but major galactoside is galactose. Lipoproteins. Lipoproteins, these are the lipids in which prosthetic group is protein. Examples are chylomicrons, very low density lipoproteins, low density lipoproteins, and high density lipoproteins. Now these are basically classified on the basis of the presence of the protein. More proteins are present, more dense will be lipoproteins. So chylomicrons have less proteins as compared to all. Very low density lipoproteins have more proteins as compared to chylomicrons. There is another class between very low density lipoproteins and low density lipoproteins, then low density lipoproteins, which contains proteins more than the very low density lipoproteins. There is another class between low density lipoproteins and high density lipoproteins, that is intermediate density lipoproteins, which contains more proteins as compared to the low density lipoproteins. Then high density lipoproteins, which has highest protein ratio. So this density basically depends upon the presence of the protein, proportional presence of the protein in them. So this shows the distribution of the different type of phospholipids in the cell membrane structure, and this distribution is not fixed. Some fatty acids are present in more proportion, some are present in less concentration. It depends upon the tissue. Derived lipids. What are derived lipids? These are basically lipids which are derived either from the simple lipids or from the complex lipids, or these are precursors of the lipids, means lipids are formed from it. Examples are fatty acids. We know that when we do hydrolysis of the triacyl glycerol, fatty acids are formed. So as fatty acids are formed from the triacyl glycerol, so these are derived lipids. Steroids, these are the hormones which are formed from the cholesterol. Cholesterol, it is also formed from the lipids, acetyl CoA, and vitamin A and vitamin D, they are also formed from the cholesterol. So all these lipids are examples of the derived lipids, as these are formed either from the simple lipids or from the precursor of the lipids. So most common examples of derived lipids are fatty acids, steroids, cholesterols, vitamin A and vitamin D. So general functions of the lipids. Lipids, it is the most abundant storage form of energy. So here I want to clear one point. Lipid is most abundant form of energy, but carbohydrate is more readily available form of the energy. Secondly, this lipids are abundantly present in the cell membrane. They are precursor of steroid hormones, vitamin D. They act as thermal insulator. You know that under our skin there is a layer of the lipids which act as an insulator. Then they protect the internal organs from the mechanical injury. They make cushions for the organs and prevent them from the injury. They helps in the absorption of fat soluble vitamins. They are integral part of the lipoproteins which have transport function. The most important structure, which is called surfactant, which reduce the surface tension of the lungs and helps in the expansion of the alveoli, it is also lipid in nature. They improve the taste and palatability. Our taste buds, they are also made up of lipids. They act as electrical insulators. You know that the neurons, the fast conducting neurons, they basically are made up of, they have a sheath around them which is called myelin sheath, which helps in the conduction, which is lipid in nature. So this is all about the fast or the quick review of the chemistry of the lipid along with the classification.